In this class, we are going to study about the fourth category of the kingdom Protista, and that is the slime molds. So these have been named as slime molds because these have something called a slimy layer about their exterior surface, on the exterior of their body. So that is why these are called as slime molds. Now these are also called as false fungi, because normally it is regarded that fungi has been originated from this group of Protista. But if we study about the evolution of these categories in detail, then we find out that the next category of Protista, which is called as Protozoa. So actually Protozoa has a category called Sarcodines. So actually these sarcodines are more related to the slime molds as compared to the fungi. But normally these are also called as false fungi. So it gives an impression that it is very much related to the fungi kingdom. So it's obvious that fungi is related to it, and it may be that fungi might have originated from this category, but normally the protozoans of the Protista are more related to this group as compared to the fungi, and we will study about that in a bit more detail in the chapter evolution. So this is the false fungi or the slime molds, and the reason behind its name is the slimy layer on the exterior surface of these organisms. So these are also normally unicellular, but we will see later that a multicellular form called Pseudo Pseudoplasmodium occurs here, but normally these are also unicellular. And the other thing to notice about these is that these are saprophytic. So we have seen that the first three categories of the kingdom Protista were the dinoflagellates, then the euglenoids, and also the chrysophytes. All these three categories were alga-like, or these were the photosynthetic autotrophs. But this category is a sort of Protestant fungi. So apart from slime molds, false fungi, these are also called as Protestant fungi because these are decomposers, these are saprophytes just like fungi. So we know fungi has a decomposer role. Apart from that, that is also a heterotrophic category, and this is also a heterotrophic category, and in heterotrophic this is a saprophytic category. So it is very much similar to fungi in those respects because it is also a decomposer and a saprophytic. So it is normally found in moist habitats where lot of moisture is found. Apart from that, some aquatic forms are also found. So the majority of these organisms live in moist conditions but not exactly aquatic. But a small category of this group belongs to the aquatic habitat. And wherever it is found, it is normal that that area will have certain decaying plant matter. So these are saprophytic, and these are normally found in moist conditions and also where lot of dead plant material or decomposing plant material is found. So this is about the major features of this category, and that is these are saprophytic protists develop a slimy mass. So because of this slimy mass, these are called as slime molds. And now during their vegetative phase, so vegetative phase is the phase during which this group undergoes growth, or it shows its vegetative growth, or the normal body size increases where it obtains its nourishment and increases in size. That is the vegetative phase. So during that phase it develops a slimy mass. Now these are normally found in moist habitat, but some also are found in aquatic habitats, and in both the habitat there should be some decaying plant matter. Normally it could also be found on decaying animal matter or some other organisms dead remains, but normally or the majority of this group's category is found near decaying plant matter. That is why it is called as saprophytic. So wherever this phytic word comes, we know phytic means something related to plants, and saprotrophs or the saprophytic means it feeds on dead and decaying organisms. So saprophytic, it is saying that it lives on decaying plant matter normally, but it can also be found on other decaying matter of other kingdoms categories. Now their somatic body, this is the main body, or we know that protists are unicellular eukaryotes, so obviously instead of body we can say the cell. So their somatic cell are either uninucleate mixamoeba or multinucleate plasmodium. So these have either uninucleate mixamoeba, and we will later study that this multinucleate protoplasm is called as plasmodium. So we are going to later study that over here. So this is found in a type of slime molds. So this multinucleate protoplasm that we are talking about, it is actually a form, and that is called as plasmodium. So it is normally occurring in two forms of somatic bodies, and that is mixamoeba, which is unicellular, or it is also uninucleate, it has single nucleus. But the other form, that is the acellular slime molds, they have their somatic body as the multinucleate cytoplasmic or protoplasmic structure, which is called as plasmodium. So this is the difference between these two forms of the category. So these are uninucleate, these are multinucleate, and we will later classify into these two categories. Now the next point is that slime molds have very similar traits to the next three kingdoms. So after the after the kingdom Protista, we are going to study about plant kingdom, animal kingdom, and the fungi kingdom. So this category, that is slime molds, has similar characteristics to the plant kingdom, and also to the animal kingdom, and also to the fungi. So these have some traits similar to that. For example, if we see the plant-like traits, then they have cellulosic spore coat, and we know where cellulose is found. So in plant kingdom, cells are normally covered by the cellulosic cell wall. So this cellulose presence is a characteristic similar to the plants. Now these also have some animal-like characteristics. So in animals, we know that animals do not possess cell wall. Animal cells only have the cell membrane. And here also their main phase, that is the somatic phase, that is their main phase. During that phase, these do not have cell wall. So this absence of cell wall during the somatic phase is an indicative characteristic of the animal-like feature. So we know that animals do not possess cell wall. We only have the cell membrane. So this characteristic is similar to the animals. Now this also has been called as false fungi. Then it is for sure that these must have some characteristics similar to the fungi, and the characteristic which is similar to the fungi is the spore formation method, or the mode of spore formation. So in the next kingdom, that is the fungi, we are going to study that there are lots of various different type of spores, like conidia, or the oidea, or the sporangiospores, or the chlamydiospores. So these are the certain types of spores that are found in the kingdom fungi. So here also the mode of spore formation is similar to fungi. So this is a fungi-like category. So from here we can conclude that this group has certain features which are similar to the next three kingdoms. But more than these three kingdoms, this is related to the protozoa, and we know that protozoa is a category of protists which we will study after this group. So these slime molds have plant, animal, and fungi-like traits. And if we will study about the characteristics of the next category, fungi, we know that the main ecological role of a fungi is decomposer. So fungi normally acts as a decomposer of dead remains of all the kingdom's categories. So this kingdom or this category also has fungi-like properties. Therefore, these will also act like the decomposers, that is, these will decompose the organic remains. But they have also a very important feature. Now, there are other decomposers also, like bacteria, fungi, and also a few other examples. So all these are decomposers, these are also decomposers, but these have other role, that is, it will keep the other decomposers population under control. For example, if at a certain place the population of decomposers increases more than required, So obviously those can decompose other living material also. Therefore this slime molds keeps the population of other decomposers, for example bacteria or the different categories of bacteria. So these will keep their population under control, because in nature everything is well balanced, and therefore the population of decomposers also should be maintained in a certain amount. So therefore these will either decompose also. Apart from that, these will also control the other decomposers like fungi and bacteria. Now there are two things that we have to remember. Above we studied about this multinucleate protoplasm. So this multinucleate protoplasm is this Plasmodium. So there are two forms. We know one form is the myxamoeba, and the other one, that is the multinucleate protoplasm, is normally called as the Plasmodium. Now these have very similar properties to the Pseudo Pseudoplasmodium, and in NCERT it is given that the Plasmodium spreads in a large area during favorable conditions, and during unfavorable condition the Plasmodium differentiates and it breaks into different pieces and results in the formation of spores, which have very thick wall, and those are very resistant to environmental conditions. So we must understand first what is Plasmodium or Pseudoplasmodium, because ultimately these are the structures which will give rise to the thick walled spores, which will act as a perennating structure. That is, the spores which will be formed by the Pseudoplasmodium or the Plasmodium, these will be resistant to environmental stress like drought or temperature or acidity or basicity or any other such feature. So this Plasmodium is a multinucleate. We know from above that this is a multinucleate. Apart from that, it is also unicellular or acellular. So acellular normally means that these do not have all the properties of a normal cell. So either this Plasmodium could be unicellular or acellular, and from above we know it is multinucleate, and it occurs in a category of slime molds. There are two types of slime molds. One is called as cellular and the other is called as acellular. So it occurs normally in acellular slime molds. So this is the property of acellular slime molds which we will study here. We have to study about these two categories of slime molds. So this was about the Plasmodium. Now we have another similar structure, or these have certain properties similar to the Plasmodium, and that is the Pseudoplasmodium, and it is a multicellular. So comparing it from above, we can see that it is unicellular, it is a multicellular, where individual cells are uninucleate. Now it is a different feature. Above we saw that the Plasmodium is a multinucleate feature or multinucleate structure, but if we see this as a uninucleate structure, so it is multicellular, but every cell of that multicellular structure is uninucleate. So on the basis of number of nuclei, we can say Plasmodium is a multinucleate structure, whereas the Pseudoplasmodium is a uninucleate structure. So this is a unicellular, this is multicellular. That is uninucleate, this is multinucleate. Sorry, that is a multinucleate and this is uninucleate. Now here is a very important feature. When we normally see that it is a multicellular structure, we normally confer or we normally think that it is a multicellular form of slime mold, but it is not so. This Pseudoplasmodium is a community-like association. It is not a multicellular form of slime mold. It is just a temporary association of different cellular slime molds or different myxamoeba. We saw that these were unicellulate. So this Plasmodium is formed by, this Pseudoplasmodium is formed by many mixamoeba which come together, and therefore this Pseudoplasmodium becomes multicellular. So it is not exactly a multicellular form, but it is an association of mixamoeba. Therefore it is being written here that it is a multicellular structure. So if different slime molds are coming together, that means all those slime molds were individual organisms. So these have come together as a community. So this is not exactly multicellular, it is just an association of different cells. So therefore all those cells are called as independent. So all those cells which have aggregated are independent from each other. So in the cellular form of slime molds, all the cells which have aggregated, these are the normal independent of each other. These are not very much dependent on each other. Now it is found in cellular slime molds, and that was found in acellular slime molds. So we will see here that mixamoeba or Pseudoplasmodium is occurring in cellular slime molds, and this Plasmodium is occurring in acellular slime molds. Now these are the two types, or we can say the two categories of slime molds, and we have to understand the differences between these two. So instead of studying the definition and characteristics, if we directly compare these two, then we can easily understand their features and also differentiate between these two. Now the first thing about the cellular slime mold is that we saw there was an association, that was the Pseudoplasmodium. So because of that association of a community, it is also called as communal cellular slime molds. So instead of cellular slime molds, we can also say that these are communal slime molds. So this is the first thing, that is, it is an association which we will study later. Now the first thing and the first difference is that these are haploid in their somatic phase. So they have a haploid. So we know haploid means n number of chromosomes, or only the n chromosome, that is the half the number of chromosomes, and these have diploid somatic phase. So diploid means 2n. So here n number of chromosomes, here 2n number of chromosome in the somatic phase. Obviously in the zygote phase both will have the 2n number of chromosomes, but the normal somatic phase is haploid, that is n. It is diploid, that is 2n, or the normal number of chromosomes, and it is normally called as mixamoeba. We studied about uninucleate mixamoeba, so that mixamoeba is also unicellular. Now these have multinucleate plasmodium. We have already studied about the multinucleate plasmodium, and it is found in the acellular slime molds. We studied about the plasmodium right here. Now that was the first difference. The second difference is about this Pseudoplasmodium. So we just discussed that this Pseudoplasmodium was formed by normal association of many mixamoeba, and here no such association or aggregation occurs. So this Pseudoplasmodium that has been formed is a multicellular structure because various different mixamoeba have come together. Here no such plasmodiums will come together. Therefore the Pseudoplasmodium is formed here and not here. Here Pseudoplasmodium is not formed because no aggregation or no association takes place in this case. Now the third difference is that sporangia are naked. So sporangia are those structures inside which the spores will be formed. So the spore forming structure are called as sporangium, and its plural is sporangia. Now here there is no coating around the sporangia, but here there is a coating called as peridium. So this coating is present over the spore forming fruiting body, or normally spore forming structure, which is called as sporangia in plural form and sporangium in singular form. So this was the third difference. Now in the starting of this chapter we studied about life cycle of protist, and life cycle of protist we studied about two types. diplontic and haplontic. So in diplontic life cycle, we saw that the somatic phase was diploid, and in haplontic life cycle, the main body or the somatic phase was the haploid. So in both these cases, gametic meiosis and the normal zygotic meiosis takes place. So here we have to see which has a diplontic life cycle and which has a haplontic life cycle. So we can see here that life cycle shows zygotic meiosis. So where zygotic meiosis takes place, we know that is called as a haplontic life cycle. So once we write here that zygotic meiosis takes place, that means this cellular slime molds have a haplontic life cycle, and that is why we have seen it is haploid somatic phase. Why? Because it is showing zygotic meiosis. And in the starting of this chapter, we studied about zygotic meiosis. Now here gametic meiosis takes place, and in the starting of this chapter, we know where gametic meiosis takes place, and that is called as diplontic life cycle. And that is why we found diploid structure over here, because it is showing gametic structure, gametic meiosis. And where gametic meiosis takes place, that type of life cycle is called as diplontic life cycle. Now we also have studied about the types of sexual reproduction. For example, isogamous, anisogamous, oogamous. So these types of sexual reproduction we have already studied. Now we see that here the sexual reproduction is anisogamous. So anisogamous means that the male and the female sex cells which will fuse are different from each other. So here there would be something called as macrocyst formation. So what exactly happens in this case is that two mixamoeba, so one mixamoeba will become much larger, and that will function as a female gamete. So once it is a larger gamete, and it will attract many smaller mixamoeba which will function as a male gamete. So in anisogamous, there is a large female gamete and small male gamete, and that fusion or fertilization will take place and zygote will be formed, and that zygote will secrete a thick wall around itself, and that thick walled or cysted zygote is called as macrocyst. So zygote here is covered by a thick wall, and that cyst is called as a macrocyst. Here the sexual reproduction is isogamous. Isogamous means that the two fusing cells are so similar to each other that we cannot differentiate which is the male gamete and which is the female gamete. So obviously here the sexual reproduction is isogamous, and here macrocyst is not formed. Here a normal zygote is formed. So here the zygote has a cyst, that is why it is called as a macrocyst. Here the zygote does not have a cyst wall. That is why we are saying that macrocyst formation does not take place in the sexual reproduction, or rather the isogamous sexual reproduction in the case of acellular slime molds. So these were the normal and the most important differences